This is a diagram of a basic cell and its organelles. We're going to start with the plasma membrane, which is comprised of lipids and proteins that forms the external boundary of the cytoplasm of the cell. Inside the cell is the cytoplasm, which is where all the organelles are embedded in. The nucleus is the center of the cell, generally, and it's a membrane-bound structure that contains the cell's hereditary information. It controls cell growth and reproduction. It's basically the cell's control center. The nuclear envelope is the nuclear membrane, also is what it's known as, encloses the nucleus. It's made up of a double lipid bilayer with pores that allow things to go in and out. It regulates passage of macromolecules like proteins and RNA, but permits free passage of water, ions, ATP, and other small molecules. Around and a continuation of the nuclear envelope is the rough ER. It's the outer membrane of the nuclear envelope, and its cisternae adjacent to each other are connected by bridges. It also has ribosomes on it, and that's generally how you can tell the difference between the rough ER and the smooth ER. The smooth ER is actually a continuation of the rough ER, and the difference is the cisternae are more tubular and they actually lack the ribosomes. The endoplasmic reticulum synthesizes steroids and other lipids. They detoxifies alcohol and other drugs and manufactures all membranes of the cell. The ribosomes, which we talked about, which are free-floating in the cell, but also on the rough ER, are produced in the nucleolus, in the nucleus. And they read genetic material, produce peptides and proteins, and assemble amino acids. The Golgi apparatus, or complex, either or, is a small system of cisternae, and it's used to synthesize carbs and put final touches on the proteins from the ER. The vesicles, which are Golgi vesicles, bud off the rim of the cisternae of the Golgi apparatus, and some become lysosomes, some fuse to the plasma membrane to help rebuild it, and others just become other vesicles. Lysosomes are the package of enzymes, or what they're known for. They're a specialized type of Golgi body. They're small, hollow pockets, and they contain digestive enzymes. They travel through the cell and break down unwanted material, and they also aid in cell suicide. Peroxisomes are produced by the endoplasmic reticulum, the ER, and they help to detoxify the cytosol. They also use molecular oxygen to oxidize organic material, and they create hydrogen peroxide. That's where we get it from. They're especially abundant in the liver and kidney cells, and that's why they're used to detoxify things like that. The mitochondria are also what is known as the powerhouse organelle. They synthesize the ATP. They're very mobile, and they're constantly moving and changing shape, so you'll see them in different shapes and sizes. They're surrounded by a double membrane, and their inner membrane folds, and there's a cisternae in there, and that's where ribosomes are used, and that's where the ATP is actually synthesized. And the last organelle is the centrioles, which are the pretty easy one to identify because they look kind of like a star. They're assembly of microtubules, 9 pairs of 3, which is what you can see around there. They play a role in cell division, and they help in the formation of spindle fibers that separate chromosomes. So they're what helps with mitosis. They also help in the formation of the cilia and flagella on the surface of the cells, which help with movement. And those are the 11 basic organelles that are important to know when studying a cell.